Good morning, class. I hope everybody's doing well. Let's go ahead and get started on today because we have a lot of material to cover that's a little bit dense. So today I'm going to be talking about DNA replication, but before I get into that, it was brought to my attention that UCR is going to be celebrating Juneteenth this year. So this coming Monday, we will not have any class, which means we're only going to have 3 lectures next week. I will go ahead and update this on my syllabus later today, and I'll go ahead and upload that onto iLearn. So before I also get into that, somebody asked me a question yesterday that I wasn't sure the answer was. And the question was, are plasmids small copies of the nucleoid? And after looking up the answer is no. So these plasmids over here are separate from the nucleoid. And the plasmids actually come from either the external environment that the bacteria is just able to pick up because there's DNA really everywhere, or it exchanges it between other bacteria during conjugation or bacteria's version of sex. The nucleoid information is only transferred to other bacteria when the cell is actually dividing. However, when a bacteria divides, these plasmids may or may not get transferred to the daughter cell. So hopefully that cleared up any questions about that. So let's go ahead and get into today. So the outline for today, I'm going to be talking about the structure of DNA. So this is going to be the main topic of this lecture and the next. Basically, today we're going to be talking more about what the DNA looks like and how it replicates. And then I'm going to go ahead and cover some important people that discovered DNA and some of their findings. The way that I'm going to structure this lecture when it comes to DNA replication is I'm going to put it, I'm going to tell you guys the stuff in layers because it is really dense material. So I'm going to talk about some of the key players, so some of the enzymes that are involved in DNA replication. And then from after we understand who the key players are, we'll go and actually talk about the steps between them. And then I don't think it's just important to know like this whole process of DNA, but I want you guys to also understand the implications of DNA and its compositions to health. Specifically to cancer. So that'll be how I'm ending the lecture. Let's see, I'm getting some questions. Oh, someone already answered it. Okay, yes. So again, if you understand my learning objectives, and I just realized I had an error in it, leaning, you're gonna do fine on the exam. So what I really want you guys to take away from today's lecture is understand what are the DNA building blocks. So this has to do with the nucleotides that make up DNA, but also how they bind to one another, both their backbone and their base pairs. And I'll get more into that in next couple of slides. I want you guys to know the 4 cell cycle phases. So I'm sure you guys have all heard of mitosis in your high school classes, biology classes. I'm not going to go over mitosis, but I want you to understand basically how the points that a cell needs to undergo in order to actually replicate and divide. I want you to understand this concept of 5' to 3' direction, because that's an important concept when it comes to how DNA is read and how some of the enzymes function. And I want you to know the steps and the enzymes that are involved in DNA replication. And so I won't go as in depth as I would in an upper div class, but I will highlight key enzymes and key steps that I do want you to know that I will end up quizzing you guys on. And then finally, I want you to understand the implications of telomeres and what implications that has to both their health and disease. And I found another error. All right, so DNA holds the instructions of all living things. It's what makes us us. And DNA is the shorthand for deoxyribose nucleic acid. And it's important to note this structure of the DNA. It has this double helix, and it's pretty important when it comes to having that shape, when it comes to being condensed within the actual cell. The DNA is made up of a series of nucleotides. And what these nucleotides are consisted of are three main parts that you guys should all know. So the first part is a 5-carbon sugar. And so the name of this 5-carbon sugar is pentose. And in DNA, the specific name for this pentose is deoxyribose. And this again is only for DNA. And this is what the typical structure looks like. So if we break down the word deoxyribose, ribose referring to the type of sugar that it is, and deoxy without oxygen is the reason why this name has the name it does. So if we look at this carbon that has this hydrogen attached to it, it lacks an oxygen where some other pentose molecules actually have this, and those usually will just be called ribose. And that'll be important to note, and I'll reemphasize this tomorrow when we're comparing DNA to RNA. So the other part that you really want to consider is the phosphate group. And so the phosphate group is attached to the ribose on the left-hand side. And then on the right-hand side, we have a nitrogenous base. And so with DNA, we have 4 different base pairs. And these are all 4 that I want you to know and understand, because you will get questions, which we'll have a couple of practice ones in a bit. But basically, adenine, thymine, guanine, and cytosine are all the 4 main letters that make up DNA. And so what you guys need to understand is how these base pairs bind to one another. So they each have only one binding partner. So adenine binds to thymine, and guanosine binds to cytosine. And so it's also important to note how these nucleotides bind to one another. And that's through this, this bonding called hydrogen bonds. And so what that means is when the positively charged hydrogen atoms, as seen here, end up binding weakly to the electronegative atoms, such as nitrogen or oxygen. And so if you guys took high school chemistry, when we're talking about electrons in the outer shell of the atom, basically there's a couple of free ones that are found in nitrogen and oxygen. And these will have some kind of negative charge. And so this negative charge will end up interacting with these hydrogens and form this hydrogen bond. So this is a key type of bonding that I do want you to know. So if I ask you what kind of bonding exists between base pairs, then hydrogen bonds is the answer. Here's water molecules undergo hydrogen bonding as well. So I just have this just to reiterate the point, where we have an electronegative oxygen that has 2 hydrogen attached to it, and the oxygen weakly binds to the positively charged hydrogen. And so this is how, you know, water molecules interact with one another, and why water is so fluid. So when nucleotides are bound together, they're called polynucleotides, poly meaning many. And so here we have an example of what a polynucleotide sequence looks like. So what you need to understand is basically how they bind to one another. So base pairs end up binding with other base pairs across, whereas the actual nucleotides bind to one another through the phosphate and the sugar groups. So basically the bonding that exists between the phosphate group and the sugar, or ribose, is through this phosphodiester bonding. And that's basically the hydroxyl groups from the sugar interacting with the phosphate group. And so these bonds form at a 5' carbon first, and then a 3' carbon. And what I mean by that is how the carbons are labeled within a structure. And so basically, when it comes to like organic chemistry, they have a whole series of nomenclatures when it comes to labeling certain things. And so when it comes to labeling carbons, they basically are carbons in a sugar. They start from where the base is located, and each one of these has a number associated with it. And so when it comes to a nucleotide binding to something, basically the nucleotide before this nucleotide here is going to bind to the 5' carbon, as we can see here or here. Now, the nucleotide that follows the previous nucleotide will bind to 3' carbon, as we can see here and here. And then here's another diagram just depicting where those 5' and 3' carbons lie. And so this is where we get this concept of DNA being read in a 5' to 3' direction. It has to do with how the nucleotides are bound to one another and the location of those specific carbons. So the primary structure of DNA. So I kind of went over what a nucleotide looks like, but let's go ahead and go a little bit more depth in terms of how all of it interacts with each other. So again, this is the image I had of a previous nucleotide. So the backbone that makes up DNA are the phosphate group and the sugar. And then after that, you basically have a nitrogenous base sticking from the backbone. And so to kind of get this point across, use the analogy of a ladder. And so basically the side rails are going to be where the backbone lies. And that's going to be, again, the phosphate group and the sugar. And so this is what's interacting with each other when it comes to the DNA nucleotides binding to one another. And again, DNA is double-stranded. So here we have 2 strands that exist here, and the phosphate and sugar groups are bound to one another. Now, when we look at how the nucleotides bind to one another, the base pairs, we see that they're analogous to the steps in the ladder. And so it's these nucleotides that are binding to one another, and that's through, again, hydrogen bonding, and these are through phosphodiesterase bonding. So let's see if you guys can go ahead and build a DNA strand. I'm going to go ahead and give you guys 2 minutes to go ahead and answer these, and then let's go ahead and check to see if people are having a good understanding of this. So I'm going to go ahead and set a timer and go ahead. Somebody asked me if we're entering this solely. Yes, so just answer it by yourself, and then we'll go ahead and check. I want to take a poll to see if you guys all got the right answers. I'll take a poll right now. You don't need to type it in the chat. If you want to, by all means. All right. So let's see. Did you guys all get this answer for question 1? I should double check this. Okay, the majority of you guys have answered. So the majority of you guys did get it correct, or you guys had an understanding, so that's good. So there were 4 people that didn't understand or had some problems with it, which is okay. I will go ahead and explain really quick. So basically, if you go back to my slides before, certain nucleotides have specific base pairs. So a T will always bind to an A, and an A will always bind to a T. And with a C will always bind to a G, and a G will always bind to a C. I hope that cleared up the confusion. I'm gonna go ahead and see how people did with the second one. All right, again, the majority of you guys have answered. So there are still 2 people that are having trouble with this. If you guys feel comfortable, do you mind telling me what you guys are struggling to understand with this? And if you guys are shy, we can go ahead and discuss it after class or during my office hours. But let me know in the chat if you want to ask or help me understand what you're confused about. All right, we got shy people. That's okay. Just come see me after, and I will be more than happy to explain what your confusion is. Okay, good. So I'm never gonna ask you guys to memorize people, but I do think it's important to give credit where credit's due, as this is where these are the pioneers of why we know what we know. And so the first person I want to acknowledge is Friedrich Nietzsche, and he's a Swiss physician and biologist. And during his studies, he was trying to understand what is within the nuclei of white blood cells. So white blood cells is just an umbrella term for immune cells. So any immune cell will fall under this category. And so he was actually the first person that discovered DNA in 1869. And he did this by collecting bandages from hospitals that contained pus. And what he did with this is he digested away all the fats and the proteins and was left with this snot-like material. And so he rationalized that there was something else other than just fats and proteins. And so we ended up calling this material was nuclein, and we now know this as nucleic acids. And so a fun fact, the pus that he was actually collecting from bandages is actually from an immune cell called neutrophils. And so I'll talk a little bit more about them in the immunology section, but I just feel it's an interesting fact to know, because basically when, say, you cut yourself and you end up introducing some bacteria into the wound, neutrophils will be one of the first immune cells that will come and basically blow themselves up. And they do this by basically releasing all of their DNA that's within them to surround all of that bacteria so it can't spread and go to other parts of the body. Let's see, some chat in here. Okay, I think somebody is hoping. Thank you. And if you still have confusions with the question, again, come see me afterwards. So the other person that I want to give credit to is Rosalind Franklin. And I don't know how many people have heard of her, but she was an English chemist and an X-ray crystallographer. And she was the first true person to confirm that DNA has the helical structure. It wasn't Watson and Crick who ended up basically using her work to publish their own. So usually it's them who get the credit for discovering DNA or discovering their structure, but it wasn't. It was Rosalind Franklin. And so here is the actual famous picture of the diffraction of DNA. And so I believe it was one of Rosalind's grad students that took this picture. And so it's known as Photo 51. And she was a brilliant scientist, and she was the one that discovered that the sugar phosphate backbone actually belongs on the outside of the structure of DNA. She literally gave her life for science. Because of all of these X-ray exposures, she was exposed to high levels of radiation for the duration of her work, and this led to her dying from ovarian cancer in 1958. Very sad. So why does DNA even need to replicate itself? So for a cell to reproduce or divide, it has to copy and transmit all of its genetic material to its progeny or daughter cells. And so to do this, DNA has to replicate. And so basically here I have a diagram of the cell cycle pathway. So we start off at the G1 phase. So cells need the right signal to signal so they can divide. If they don't have that signal, then they're not going to divide. Once the cell receives that signal, it will enter this G1 phase. And during this time, basically all of the organelles are doubling except for the DNA. It's not until the S phase, which we're going to get more in depth about how this happens, is where DNA ends up getting synthesized and doubling itself. And then it's the G2 phase where the cell prepares to divide. And then the smallest phase of all of this process of a cell prepping to divide is mitosis, which is just when the actual steps of the cell actually separates from the other. And again, we're not going to cover the steps of mitosis like prophase, anaphase, etc. Just understand these different phases that occur and what's going on in each one. So before I get into DNA replication, we have to understand this concept that different cells replicate at different rates. And it's really all dependent on the signals that they get and the kind of cell that they are. So for example, hair cells or skin cells or intestinal cells, those cells are constantly dividing because they're constantly shedding and needing a new layer of these respective cell types. So they're going to be going through that cell cycle process a lot more often than, say, a neuron or some kind of muscle cell. And it's really important to know and the implications of some of this, because the fact that neurons divide very infrequently, if at all, can manifest itself in some injuries. So one of the reasons why, say somebody ends up getting a spinal cord injury and ends up being paralyzed, well, the reason why they can't really heal is because these neurons that are present here they don't really divide. And so if they don't divide, then you can't heal yourself. And so scientists are trying to work on ways for regenerative medicine, where they're trying to promote the growth of neurons, but it's a very fine balance because you don't want neurons overgrowing. So there's a lot of regulatory mechanisms that inhibit neuronal growth. So I'm going to go ahead and layer DNA replication just to kind of get a lot of these concepts across, because again, it is very complicated. And if you guys have any questions at the end, feel free to ask. So the whole point of DNA replication is to generate two daughter cells. And so it all starts with this parent strand. And this parent strand is going to serve as the template. Once the DNA is unwound and separates from itself, another typo, then the DNA nucleotides will end up being filled in. At the end of DNA replication, you're going to get two daughter strands that will consist of one parent strand and one daughter strand that's connected to that parent strand. So the general overview of DNA replication. So it begins with those two parent strands of DNA. As I was talking about yesterday, DNA is very long. And so if it had only one point where it was going to replicate, it would take freaking forever to actually, double itself. And so there's certain points within the DNA that are known as points of replication. And so the actual process of DNA replicating within a certain region is known as a replication bubble. And so basically, there's a lot of points of origin that exist within the chromosome that all of this is occurring, so it can go ahead and generate two new daughter strands, and the DNA can be copied. So before we actually get into the steps of DNA replication, we should really understand who the players are so you guys don't get confused. And so there's definitely many versions of some of these enzymes, such as DNA polymerase. There's like 3 different versions. I think DNA ligase are like 2. I'm not going to expect you guys to know that stuff. I just want you to understand the function of these enzymes and what role they play when it comes to the replication process. So it's important to note in biology, any word that ends in "ase" is typically going to be an enzyme. And so the whole point of enzymes are to speed up reactions for building or breaking things down, which we're going to see with some of these enzymes. So the first enzyme is going to be the helicase. And so once the point of origin for replication is initiated, the DNA helicase is basically going to separate the 2 strands. As I mentioned before, that there's hydrogen bonding that exists between these nucleotides. To prevent these hydrogen bondings from reforming, there are these single-stranded binding proteins that actually anchor the DNA up and keep it open. And this is really important to prevent the closure of the 2 strands. Oh, and then here I have an actual image of what a helicase looks like. And throughout my lectures, I'll have pretty pictures of what some of these molecules look like, because I think they're very interesting. The other enzyme is going to be topoisomerase. And the whole point of topoisomerase is to relax the DNA. So as the DNA opens up, and this is going to be known as the replication fork, where the DNA is just continuing to unwind itself. Above or downstream of this, the DNA ends up becoming really tight, really supercoiled. And this leads to an increased tension in the DNA. And that's not really good for it, because there's going to be a point where the DNA can't open anymore because there's so much tension downstream of the opening of the DNA. And so that's where topoisomerase comes into play. And so here we have a supercoiled DNA section, and topoisomerase basically comes in and has this ability to cut a couple of pieces of the DNA, straighten them out, and then stick them back together, or ligate. So it relieves some of that tension. And in my notes section, there's actually a nice video that kind of shows or demonstrates this with 2 coils and what happens when you separate the 2. So if you struggle with this concept of what topoisomerase does, feel free to watch that video. It's really good at explaining that whole process. The other really important player is DNA polymerase. So DNA polymerase is important for synthesizing the new daughter strands. And so what it basically does is it takes the free-floating nucleotides that are within the cell and end up putting the complementary strand on the template strand. And so it's really important to know that this polymerase moves in only one direction, and that's from the 5' to 3' direction. And this is going to be important to note when I get more into the types of the 2 strands and what's going on when nucleotides are actually being loaded onto the strands, but understand that this only moves in one direction. Another important function that DNA polymerase has is this proofreading capability. And so as the DNA polymerase is adding on nucleotides, sometimes it makes mistakes, but it's very rare where it actually does. Basically, for every 10 billion nucleotides that DNA polymerase is adding on when it's synthesizing a new strand of DNA, it only makes one mistake. And it actually has this ability to read that mistake and fix it. Considered both a building and breaking. Yeah. Somebody asked me, would topoisomerase be considered a building and breaking enzyme? Yeah, definitely. I didn't show. Actually, do I have it in here? No, I didn't put an image of topoisomerase, but if you actually look at its like structure, it actually has two parts to it. One has that cutting capability, and then the other one has that sticking capability. All right. And so the other enzyme that's really important to understand is primase. So primase here in pink basically adds on a short nucleotide sequence that is called a primer. And basically this is going to be the starting point for DNA replication. So in order for DNA polymerase to do its job, it needs to know where it begins. And so once the primer is on, then DNA polymerase is able to attach itself and then start synthesizing that new daughter strand. DNA ligase acts as glue. So the word to ligate in science just means to stick together. And so during the replication process, both of the strands of the template parent strands are being duplicated. And so each one of those new strands that are being synthesized have a specific name. So the strand that we see here is known as the leading strand, and the other strand is known as the lagging strand. And I'll go ahead and get more in depth what this means once we actually cover the steps of this whole process. But just note that in the lagging strand, basically instead of being continuously synthesized like the leading strand, it's actually made in small fragments. And because these fragments are generated and there's these little gaps that are between these fragments, DNA ligase's role is to stick these together. So now let's actually talk about the process of DNA replication. So I covered some of it when I was talking about the enzymes, but let's go ahead and go step by step. So the whole initiation process. So once the cell gets the information, okay, it's time to divide. It goes through that cell cycle process. And so once it makes it to the S phase of that, that replication diagram I showed you earlier, it's going to get the signals to go ahead and open up. So there's going to be a point of origin, and there's going to be specific sequences within the DNA that say this is where you begin to replicate. And so then what's going to end up happening at the point of replication are the helicase going to come in and basically unwind the DNA. And then again, in order to prevent the closure of those nucleotides from binding to one another, these single-stranded binding proteins come in and basically hold the DNA open. And then the primase ends up coming in and basically putting in that short primer, that small set of nucleotides, so that the DNA polymerase knows where to go. And just like the DNA polymerase, primase only moves in one direction. So we can see this is the direction of replication. And we can see that the primase is synthesizing the primer from the 5' to 3' direction. And so we can also see that this 5' to 3' direction is also occurring during in this second sequence here. And notice how this primer or this enzyme is going to this opposite direction of where this point of replications occurring, which is going to be important in understanding what that means when I talk about a lagging strand. And then we're also going to have topoisomerase recruited downstream of the replication point to relieve that tension from when the DNA is opening itself up. So again, there are both of the strands, the parent strands are being copied, but one is known as a lagging strand and one is known as the leading strand. Both strands are going to have a primer attached to them. So DNA polymerase knows where to go. Once DNA polymerase actually binds to these primers, they begin synthesizing the new strand. And so the leading strand is the easiest strand because it's just a continuous formation of one of the daughter strands. And so again, it moves from this 5' to 3' direction. And so as this DNA continues to unwind in this direction, the DNA polymerase can just keep moving and adding on nucleotides so the replication process continues. With a lagging strand on, however, because DNA polymerase moves in that one direction, it basically will make a new fragment, and you don't need to know what an Okazaki fragment is for the test. I will go ahead and briefly say all it is a combination of the primer and the new synthesized fragment. Dr. Okazaki was the one that discovered that this lagging strand process occurs where it's in fragments that occurs. I'm not going to quiz you on any of it, but if you do end up watching some videos on how DNA replication occurs, they will end up mentioning it. So just so you're aware and you don't get confused. So basically this whole process of a primer attaching itself to the lagging strand and then DNA polymerase making a fragment, and then again, the primase has to come in and add another primer, and then the DNA polymerase moves in that direction. As this continues to open up, there's this whole process of more fragments being made, and you can see why this is known as the lagging strand. And so the DNA ligase, again, the whole function is to stick DNA together. And so it really functions on the lagging strand. As I was mentioning how these fragments are generated, there are points within the DNA that aren't stuck together, and it's really specific for the backbone. And so basically what DNA ligase does is scan the sequence, and anytime there's a break within the backbone of these fragments, it just ligates it together, it sticks it together, so we have a continuous daughter strand. Let's see, I got a question. Single-stranded binding proteins is what SSB stands for. And again, those are the ones that are going to be important for when helicase unwinds it. They're the ones that anchor the DNA open so the whole process can begin. And so once the DNA is done replicating, there's a whole termination process. And so DNA replication really ends when it reaches other replication bubbles. As I mentioned before, there's multiple points within the DNA that are replicating at the same time. So once they all kind of come together, then termination of that replication process is done. But we have to, what I really want you to focus on is when this process actually ends at the site of the chromosomes, or at the end of the chromosomes. And so the names of the ends of these chromosomes are called telomeres. And so basically there's this whole process of once the DNA is about to end, when it's replicating, there's actually a long repeated sequence of nucleotides that don't really encode for anything. And their whole process is to help correct that process because the polymerase doesn't really know where to end. And so the telomeres basically will kind of use that indication like this is where you need to end, this is where replication ends. But because there's this whole correction process that occurs, every time a cell divides, the telomeres get shorter and shorter because they cut out a small sequence from the telomeres and end up ligating it back together. And so you would imagine after a few rounds of replication and your sequence that encodes for the telomeres runs out, the cell can no longer divide. It's basically a finite amount of times that it can divide. And so this is really important when it comes to talking about telomeres and our health, because every time that the cell is replicating, the ends of the chromosomes are getting shorter and shorter. And scientists now know that this plays a role in how we age. And so it's all part of the normal aging process. So if you look at a person that's young versus somebody that's old and compare their telomeres together, somebody that's older is going to have significantly shorter telomeres than somebody that is young. And so there's a lot of aging and longevity research that's focused on trying to keep these telomeres long so we can basically look young forever. I don't know the status of where they're at with this, but it is a focus for scientists. Now, when we talk about telomeres in the context of disease, basically for patients that have progeria, or another name that people commonly call it is Benjamin Button disease, their telomeres are very short when they're born. And this has to do with a genetic mutation in one of the proteins that regulates telomeres. However, with cancer, it's a little bit different. Cancer cells have abnormally long telomeres. And as I mentioned before, every time a cell replicates, it loses a piece of the end of the chromosomes. But with cancer, they have machinery that we're still trying to understand that keeps those telomeres long. And that's why basically cancer cells can divide infinitely. Now, I want to go ahead and come back to this checkpoint diagram and relate this to cancer. So again, all the cell has, when a cell is dividing, basically it's undergoing this process. But what I didn't mention before is that there's actually checkpoints within this whole process to make sure that the cell is healthy and viable. And so at the end of G1 phase, you have the first checkpoint. And basically what the host machinery is going to look at is to see if the DNA is okay, and if the cell has all of its right nutrients and particular growth factors that are going to help it continue into the next stages of replication. The next checkpoint is the G2 checkpoint. And so, we replicate our DNA, and then once we make it to G2, the cell has machinery that's going to check, did the DNA actually replicate okay? Are there any errors? Does it look fine? All right, cool. Now, what about the cell size? Does it have all of its organelles? Does it look pretty healthy? Is it good to divide? And if it is, then it's going to go on to the mitosis phase. And so again, the mitosis phase is the actual process of the cell splitting into two. And so the M phase checkpoint basically checks to see if the chromosomes are attached to these things called spindles. And what these spindles do is basically once the DNA has doubled itself, once the cell actually replicates, these spindles are going to pull these the copied versus the so the old versus the new DNA to each respective cell. And so if the spindles aren't attached to the chromosomes properly, then there's going to be problems with the cell. And so if there's any problems at any of these checkpoints, the cell's pretty good at trying to repair itself. But in the event that it can't, if the mutation or whatever is too far gone or it's too damaged where it can't be repaired, then the cell undergoes programmed cell death or apoptosis. And so this is important when we relate this to cancer, because cancer has a lot of mutations at these checkpoints, and a lot of them are the proteins that regulate these checkpoints. Let's see, I think I got a question. Oh, somebody else. Nice. So when we relate this to cancer, basically these DNA mutations lead to something called the hallmarks of cancer. And so this diagram was created from a very famous, like I think the most famous cancer biologist of our time. He discovered a lot of the genes that play a role in cancer spreading. And his name is Robert Weinberg. And so I know since then this has been updated, but just for the simplicity of the class and the events of this diagram, he basically said that there are 6 different things that a cancer cell needs in order to actually become a cancer cell. And so I'll go ahead and walk you through each of these. And if you guys want to refer to the paper where this is at, and you guys want to know more about this, I have a reference here. So we'll start off at the bottom. So a cancer cell needs to have the ability to replicate infinitely. And this happens when a lot of those checkpoints don't exist or are mutated to a point where the cell can continue even though it has a lot of DNA damage. The other thing a cancer cell needs to survive is this process called angiogenesis. And so angiogenesis is just a fancy word for making new blood vessels. And so like the blood vessels in us, cancer cells basically create them. They stem from the actual site of the tumor. And once the blood vessels are formed, it can go ahead and get nutrients, be it oxygen or sugar. And the more blood vessels the cancer has, the more it's able to grow because it's getting the nutrients it needs to grow. A cancer cell needs to be able to resist cell death. And so going back to those checkpoints again, if in a normal healthy cell, if a cell is damaged at one of those points, it will be programmed to die. But with the cancer cells, because they have a lot of mutations at some of these points, they're not able to actually die. And so they're able to basically continue. A cancer cell needs to have the right proliferation signals. And so proliferation just means to divide. And so as I mentioned before, every cell, healthy or cancer, needs some kind of signal to tell it to divide. And so if a cancer cell doesn't have this information, then it's not going to divide. But cancer being how aggressive it is, it actually can produce its own growth factor, so it can signal to itself to grow. The other thing that a cancer cell needs is to be able to avoid growth suppressors. So I didn't cover any of the machinery that plays a role in checking or regulating some of these checkpoints, but there are these proteins called tumor suppressors that exist within us. And basically, these proteins play a role on putting the brakes on cells. If they're growing too fast because they have any damage, these will stop the cell from growing, and these play a role in those checkpoints. Well, with cancer cells, they actually, a lot of them have mutations in these proteins, and these proteins basically can't do their job. And so that leads them to basically grow infinitely. And then the last thing the cancer cell needs is to be able to invade and metastasize. And so what this means is every cell, and I didn't cover this in the last lecture just for simplicity, but every cell has something called an extracellular matrix. And this matrix basically limits how big the cell can get. And so with cancer cells, they actually have enzymes that degrade the matrix. So if they don't have a matrix, then they're able to kind of grow and invade neighboring tissue. And so, that's how a tumor forms. That's the initiation. And so you have 2 kinds of tumors. You have benign and malignant. So you have ones that more or less just grow but don't cause any kind of disease unless it's, in your spinal cord or something, and that, can lead to paralysis. Or you have the malignant kind that basically has this ability to spread throughout the body, or the fancy word is metastasize. And so with cancer, it's important to understand that when somebody has cancer, it's not the tumor itself that leads to the patient dying, it's that tumor spreading to other parts of the body and then making more tumors and then repeating that whole process until a lot of the there's a lot of tissue damage in a lot of vital organs, and that's what leads to a patient dying. So know a couple of these for the exam. I think it's important to understand, as cancer is the second leading cause of death in the United States. I don't know if that's changed since COVID. I'm sure some of the numbers have changed, but I think it's an important concept to know, because I think we all know somebody, a friend, relative, or somebody that has or had cancer. Yeah. So let's go ahead and do another couple of practice questions, because I think I really want you to understand this concept of 5' to 3' direction. So if I were to give you a diagram as so, and you know that DNA polymerase works in a 5' to 3' direction, at which end of this parent strand is the 5' located at? Is it D or is it A? And then go ahead and answer the same for question 4. Let me go ahead and bring up that poll and see. All right, so just go ahead and answer when you feel confident in the answer. And let's see if I can. So, and I'll go ahead and answer some of the questions I'm seeing in the chat. So somebody asked, in a sense, that the cancer cells are eating our blood vessels. So no, they're not eating it. They're making their own blood vessels. So blood vessels are, it's a set of cells that link to our cardiovascular system, where, think of it as a pipe. They're making pipes to get into our blood, to get access to our bloodstream, so our blood can nourish them, because our blood has a lot of nutrients in it. So they have certain proteins that they can, or signaling molecules that they can make to make their own blood vessels, so that they can get their nutrients. Oh, and somebody answered that. All right, nice. All right. I'll give you guys like 15 more seconds. All right, and pull. So it seems like quite a few people are having trouble understanding this concept of directionality. And that's okay. Trust me, when I was here, I definitely struggled with it. And now that I think about it, maybe I didn't make myself clear when it came to answering or showing one of the diagrams. Let me go back to one of the slides, and then that might clear things up. Where did you go? Okay. So I think I'm going to update this slide just so there's more understanding for this. So how I was mentioning before that, let's go back to this slide actually. This sense of directionality with how the carbons bind to one another, right? So we start from here and we have the 5' end to going to the 3' end. What I failed to say is the so we'll start from here. So 5' end, 3' end. What I didn't say, and I'm glad that I took this poll so I could make sure that this is clear, is when the strand that's binding to this first strand, it's actually binding to it in the opposite direction. So if this is 5' to 3', then basically this over here is going to be from 3' to 5'. Does that make sense, people? I hope that makes sense. But yes, so just understand that when two strands are bound together, they're always going to be an opposite or anti-parallel one another. So we have a 5' to 3' here, then this other strand is going to be at 3' to 5'. Does anybody have questions about that? No. All right. Okay. I'm wondering, I think what I'm gonna do is do that poll again. Let's see if these results get better. Maybe I should look at other diagrams really quick here. I know. I think the more ways we visualize something, the better it is. So here we have a 5' to 3' strand. So this is going to be the template strand. Then we have another template strand here. When it's bound, it's from the 3' to 5' direction. So it's anti-parallel or opposite of the other strand that it's bound to. And so when they separate, basically, so we have the 5' to 3'. When they separate and this new daughter strand is being synthesized, it's being synthesized from the 5' to 3' direction. And same thing, 5' to 3'. So if we look at this, we see that the 5' is going to 3'. So it's going to be anytime that a second strand is being made or is present, it's always going to be the opposite of whatever the other strand attached to it is. So I hope that concept makes sense. If it doesn't, I'm going to go ahead and take a poll again. And if people are still struggling with the concept, we can go ahead and talk after class. Let's see. Stop sharing. Oh, yes. Okay, let's go ahead and try this again. So now that you understand that these are opposite of each other, let's see if these numbers are better. All right, another couple more seconds. All right, so you guys did a lot better, so that's good. You guys are understanding that concept. For those people that are still struggling, we can go ahead and talk after class and I can go ahead and get people's confusion, and then I'll do my best to try to clear up any of that. All right, let's move on. So here I have a just summary for you guys to look at when it comes to trying to look at the different steps that occur during DNA replication. I think it's helpful to see like a big picture of everything. And then here, notice how I have labeled the respective directions. So I think what might be helpful is if you kind of follow this process and see how things connect. and look and understand how these enzymes move. And then just use the enzymes or the strand as a reference point. So if I tell you, for example, like in the last question, that this enzyme moves from a 5' to 3' direction, and I ask you, well, what this top strand, what does it look like? You can just use the opposite of here. So we know that this is 5', oops, and this is 3'. And again, anybody that is struggling to understand this, please stay after and I will do my best to explain. I'll also have a list of a summary of the proteins that are involved in this process. This is just good for you guys to study so you understand. And then now we'll have our breakout session that we have for our class. So basically what I want you to do is I'm going to break you guys up into groups of 2 to 3. And what I want you to do is help each other understand this process of DNA replication. And so there's only a handful of steps that occur. So, you know, go through these different enzymes, understand what they do and when they come about. And so basically assign somebody a number. If you have 3 people in your group, assign someone 1, 2, 3. There's 2, assign 1 and 2, and then go through each one of these and say like what it does. And if somebody's struggling to understand these concepts, this is the time to help each other understand this so you guys can all do well. I'll give you guys about 5 minutes to do this. And then after, I will go ahead and open up the sign-in sheet so you can get your participation points for the day. And then we're done with class. So I'm going to go ahead and stop this really quick. And then I will go ahead and break you guys out.